Okay, let's get started with our free webinar. Welcome to Lagoon Microbiology. Meet your bugs. I'm Julie Hartwig. I'm a lagoon specialist with Triple Point Environmental. And I'm having trouble advancing. There we go. So an overview of what we're going to be doing today. And please give me a shout if you are having trouble hearing me. We'll start with an introduction to who Triple Point is and what we do if you have not been on one of our webinars before. Then we're going to go into the basics of microbiology and how it works for wastewater treatment and lagoons specifically. Next, we'll do a review of the microbes that make up the lagoon treatment. Those include bacteria that eat BOD, sludge, and ammonia. Those are nitrifying bacteria. We'll go through algae and the higher life forms that can be found in the lagoon. And we'll finish up with a review of how you can use that knowledge of bacteria to diagnose issues with the operation of your lagoon. So to kick things off, Triple Point Environmental is a lagoon services company, and our motto is that lagoons do it better. We know that this goes against some of the prevailing wisdom right now, that lagoons are obsolete or that they should always be removed and replaced, but we believe that's incorrect, that lagoons have years left of service. They're a very cost-effective and operationally efficient way to treat wastewater, particularly for many smaller municipalities or plants. So we're trying to get the word out that lagoons can meet new permits and do it better even than newer plants. So to support that, we're starting a Lagoons Do It Better community online. The first line of that is our informational blog on our website, tpenv.com. We also have educational videos on our YouTube channel. And we conduct training events around the country to teach operators and engineers how to better operate their lagoon and understand it. And we have a Facebook community, Lagoons Do It Better, where we're trying to connect operators with operators so they can learn from each other's experience. And lastly, we have free swag that if you follow us on YouTube and join our Facebook Lagoons Do It Better community, we will send you the fabulous Lagoons Do It Better camo hat. And we also have poop emoji, little foam toys that we use at trade shows. And if you'd like one of those, please send us a request for that. I mentioned the lagoon training. We've got a session coming up that we're taking registration for in Iowa in May. We'll be having two sessions, one on May 6th and 7th, and the second on the 8th and the 9th. These will be two-day on-site events where you can do not only classroom training, but also hands-on instruction in the lagoon and also our nitrox ammonia reactors. So if you're interested in that, please visit our website. And lastly, for the intro, here is an overview of Triple Point's products, our solutions. The first one for lagoons is the Mars aerator. It's an aerator that combines fine bubble and coarse bubble aeration that accomplishes two objectives. The fine bubble is very efficient for oxygen transfer and aeration, but we include a coarse bubble aerator because mixing, as we'll talk about later in this presentation, is also hugely important. And it's an important part of an aeration system, not just lagoon operation. Our next product is our nitrox reactor. As states around the country and regulators roll out ammonia limits on people's permits, lagoons are needing to meet those, and a lot of the conditions in the lagoon itself are not suited to that. So we created a lagoon ammonia removal reactor. It's a full flow reactor that optimizes conditions to fully nitrify and remove ammonia after the lagoon. We have an expansion on that called the nitrox plus D process. If you do get a total nitrogen limit, this expands the nitrox reactor with additional cells to do denitrification. So that not only removes your ammonia, but also the total nitrogen itself. And finally, our PhosBox system is a tertiary filter to chemically remove phosphorus from the lagoon effluent. So to start off with the main content of this webinar, about the importance of biological treatment. It's usually a given. It's part of the design process, but we don't really usually take a step back and think about why biological treatment is so important. And that's because it's the most efficient way of removing soluble compounds from wastewater. Up until more recent times, it was functionally the only way to remove soluble things like BOD from wastewater. You can throw coagulant chemistry at it all day and you will not get it to precipitate out. The alternative to this is ultrafiltration or reverse osmosis, but biological treatment is far more efficient than that and more available and has been used for centuries. And it's the primary mechanism of lagoon treatment. You don't have filters and headworks a lot of the time. You get some settling over the course of the residence time of the lagoon, but really the reason that lagoons are able to treat wastewater is entirely because of a volunteer biological community in the lagoon. And finally, biological treatment allows for true removal of BOD particularly, and also can be used for nitrogen without waste. So if you're carbonaceous BOD that's converted to carbon dioxide and off-gassed to the atmosphere, that's truly removed from the waste stream without creating sludge. Obviously, some of it will end up in biomass and it will end up in sludge, but actually the majority of it is just released to atmosphere. And there are very few processes that allow removal that simply and efficiently without having to handle waste products. So now to get into the meat of it. Biological metabolism, if we're looking down at the bacterial level, they need energy to live and grow. A bacteria's purpose in life is to make more bacteria, and for them to live and thrive and reproduce, they need energy, and they get that by breaking down molecules in the wastewater stream. The one exception to this are algae, because they use photosynthesis. They get their energy from the sun. We'll talk about that later. But for all the other species we're talking about chemical energy. And they need energy, and we need chemicals to be removed from the water. So this is a good partnership between bacteria and wastewater operations. So as they break down this chemical energy, the currency that the cell uses to capture and retain that energy is a molecule called ATP. It's adenosine triphosphate, and that's kind of like blood sugar. It stores the energy once it's released. So the mechanism, as they break down this chemical energy, we show a tower of chemicals on the right here. This is called the redox tower. And this is a visualization of the chemical potential energy in different transformations of molecules. And each reaction that a bacteria carries out will use two of these levels. And the amount of energy that it carries depends on how far away from each other they are on the tower. The bigger the gap, the more energy that can be transferred between the two. And to illustrate this, a really powerful reaction, the one at the very top, is the oxidation of glucose, which is kind of the standard reference molecule that stands in for a lot of carbonaceous BOD at the top. And then we have the conversion at the very bottom of oxygen to H2O into water. And this is the fundamental metabolism of a BOD eating bacteria. So there you go. That is the entire span of the tower. It's a very large reaction. So you see why BOD eating bacteria are so vigorous and volunteer communities that if you have this wastewater, of course it's going to start breaking down because it's a really good source of energy for these bacteria. So as the bacteria try and create ATP, what they're doing is they're moving electrons around. That's the mechanism used to go from chemical energy to ATP, which is cellular energy. The first version of how they do this, the most efficient version, is called respiration, which means you need two separate chemical species. You have an electron donor and an electron acceptor. So the electron donor is basically the food source, and the electron acceptor is basically the waste receptacle. So for our reference molecule glucose here is the C6H12O6, and oxygen. That reaction breaks down into CO2 and H2O. And when you go and you do that, they're moving electrons off of the glucose as it's broken down, and they're sticking them on the oxygen to create water. But what happens when you have no oxygen? Then the second mechanism that a cellular metabolism can use is fermentation, which involves no separate electron acceptor. So they have an electron donor, this is their food source, but they have to somehow stick the electrons back on the original molecule and reject it from the cell as a waste product. They can do this, they gain energy from it, some through the use of enzymes, but it's a lot less efficient. So here, again, using glucose, this is the reaction that breaks it down into 2 molecules of lactic acid. And you can see that there's no waste products, but how do we really illustrate how much more efficient this is? Well, we can look at the amount of ATPs, the actual energy units that they get out of each reaction. The first one, breaking down glucose via respiration using oxygen, yields a theoretical 36 ATP per molecule of glucose. I don't know how much context it has, but that's a pretty good yield. And again, that's theoretical. They probably only get about 30 because the process isn't 100% efficient, but it's very efficient compared to fermentation, where you break down glucose into lactic acid and you get 2 ATP. So this is starting to get into why aerobic reactions and adding oxygen is so critical for treatment. Oxygen, being such a good oxidizer, allows for more energetic chemical reactions than would be available if they didn't have that really powerful electron acceptor in the water. And then looking outside of the cell metabolism a bit, all these bacteria, hundreds of them, millions of them in the environment, but hundreds of them at least in the wastewater treatment process, all of them are tuned to particular environmental factors. They've organized their metabolism around a set of conditions, and they have enzymes and everything they need to operate under those conditions. And the one that we talk about the most are aerobic versus anaerobic bacteria and microbes. But even that breakdown isn't that simple. We also have the distinction of if a bacteria is facultative versus obligate, and that's a description of whether or not a bacteria is tied to one mode or the other, or if they can do both. So a bacteria that's an obligate anaerobe can only exist, can only live in waters with oxygen, versus an obligate anaerobe cannot live in waters with oxygen. They have to have an anaerobic environment to live and thrive. And that's what this illustration is meant to demonstrate. So if you look at this as a test tube where the surface of the water contains oxygen and the bottom of the test tube does not, this is where all those different bacteria species would grow. So on the left, we have the obligate aerobe. They're only growing right at the surface. That's where the oxygen is. That's the only place they can live. There's a second class called a microaerophile. They want oxygen, but only just a little bit, which is why that band appears slightly lower under the water. And then the third one, these are the guys that can operate in both modes. These are the facultative anaerobes. When there's oxygen, they'll operate aerobically, and that's more efficient. So that's why you see this denser band right under the surface. But they can operate in both modes. They grow through the entirety of the test tube. The next type is an aerotolerant anaerobe. And these are bacteria that can live in the presence of oxygen, but they can't use it. Their metabolism is going to operate anaerobically, whether there's oxygen present or not. And that's why they are present equally all throughout the test tube, but they're operating anaerobically even in the oxygen environment. And then finally, we have the obligate anaerobe. This is a bacteria whose metabolism doesn't work right. They cannot thrive in waters with oxygen, so they're growing at the very extreme bottom of the tube where there is the least oxygen. And you can get most parts of that spectrum in a lagoon, especially a facultative lagoon will have a larger layer of anaerobic activity at the bottom of the lagoon. So you find these different bacteria in different places throughout the water column. And then two other factors that they're optimized to live in are temperature. So a wastewater environment where the water is never incredibly hot and it's liquid water, it doesn't freeze, it's not too cold. That's going to select for bacteria that like that range. But even within that range, you'll have different bacteria are peaking in efficiency at different times. A lot of bacteria like it warmer. It's more energy. Chemical reactions go faster in warmer temperatures, and that's beneficial to many species. But some species are more adapted and they can function better down at lower temperatures. And then the final one is pH. Electron transfer in and out of the cell wall is affected by pH, about the species, the chemical species that are in the water of hydronium ions, which is what pH measures. So different bacteria can only live in certain pH ranges, and municipal wastewater is generally pretty safe in the pH range. It's neutral or slightly more alkaline than neutral, which a lot of species really like. But if you get into industrial waters, there are bacteria such as, they're called acidophiles, that if your water is very acid, you can get bacteria that grow in that. But it's just one example of, there's this huge diversity of microbes, and one of my professors in grad school said that the #1 rule of ecology is that if something is available to eat, something is going to eat it. And that's what this demonstrates. No matter what the environment is, some bacteria is going to come along, specialize for it, and that's their niche in which to survive. Okay, following that, so as these bacteria are reproducing, that's their goal, they need building blocks to build cell mass, both within their own cells and then to split and create new cells. So the big building blocks are called macronutrients, and these are the bulk chemicals that are needed to create new cells. And #1, far and away for these, is carbon. That's cell mass, it's what bacteria and all life on Earth is made of. And this is usually not an issue for most bacteria in a wastewater lagoon because we have so much carbonaceous BOD, and they can eat that, metabolize it, and then use that carbon to build themselves up. The second most necessary element is nitrogen. They use these for proteins and for DNA, genetic material, as well as other various functions around the cell. And then the last one is phosphorus. That's used for nucleic acids and DNA, but also for cellular energy. You'll recall that our basic unit of cellular energy is adenosine triphosphate, and you cannot get that unless you have enough phosphorus to create those triphosphate molecules. So the general ratio that we use to describe how much of these elements that they need is a 100 to 5 to 1 ratio for carbon, nitrogen, and phosphorus. And in a municipal wastewater stream, you're going to have excess nitrogen and phosphorus compared to carbon. But this ratio and the concept of building cell mass is the reason that there is some uptake of nitrogen and phosphorus, even in a pond that's only designed to remove BOD, because they're being used, they're being scavenged for the water and built in to the mass of the bacteria itself. And if you have an industrial stream that has, say, a very high sugar content and very few nutrients, this can actually become a limiting factor in treatment. If you do not have enough nutrients, if this ratio is out of balance and something is limiting the amount of growth. And then, much like us, as humans, bacteria need vitamins, they need micronutrients. These are things like sulfur and copper. They will uptake into their cells to provide specialized role in an enzyme or a protein. So I mentioned that most bacteria on the last slide, they get their cell mass and their carbon from the food that they eat. That's only the majority of bacteria, which are considered heterotrophs. Heterotrophs versus autotrophs is a distinction in microbiology between organisms that can make their own food, which is autotrophs, and organisms that consume existing food from the environment, which are heterotrophs. And in a wastewater plant, heterotrophs are far and away the most dominant because it's a food abundant environment, and they get their carbon from the food that they eat. So it's a really good synergy. It's a strong reaction. It's a lot easier to eat other people's food than it is to grow your own from scratch. So these heterotrophs are the dominant bacteria, particularly for eating BOD. And they eat either organic molecules in the waste stream or other organisms, including other bacteria. And then on the opposite side, we have autotrophs, which are organisms that produce their own food. They still need an energy source, and that can be either sunlight through photosynthesis or chemical energy through chemosynthesis. But the chemicals they eat, since they can't use carbon from organic molecules, will be inorganic molecules. And the prime example of this one is ammonia, which we will get into later. But autotrophs, since they're making their own food and not eating carbonaceous molecules, they require a carbon source from someplace else, separate to their energy source. And in a waste stream, particularly for nitrifying ammonia-eating bacteria, a lot of them use CO2, which dissolved in water becomes alkalinity. So they take carbon from the water and energy from somewhere else and combine both of those things to build their new cell mass. Next up, we have growth kinetics. So we've discussed why and how bacteria grow and reproduce, but now looking at a more macro scale at the pond, what determines how much bacteria grow given your waste stream, a typical municipal stream or industrial stream? And we use a biological treatment efficiency equation that's based on two factors. K is a kinetic rate factor that's dependent on mixing, and T for a lagoon is time in days of treatment. And the K kinetic rates listed here are assuming that the bacteria have enough oxygen to meet their needs. And then K becomes only dependent on mixing rates in the lagoon, which can vary pretty wildly. Because mixing is necessary because a lot of these bacteria do not have the ability to move themselves around. So you need mixing to make sure that your three components here, the oxygen, the bacteria itself, and the food that they're eating, are always coming into contact with each other. So the more mixing, the more bacterial growth you'll get. And that's reflected in the kinetic rates, which vary dramatically from a facultative lagoon where mixing is only really provided by wave action on the surface, is 0.12. And then partial mix lagoons have some aeration only enough to provide oxygen without very much mixing, and that kinetic rate doubles to 0.28. But then you go to a complete mixed lagoon. This is the other end of the spectrum from a partial mix. This is enough mixing to keep all the solids in suspension. You're not going to let sludge settle because the entire lagoon is very strongly mixed. And what the effect that gives is almost an order of magnitude increase in the kinetic rate. You go from 0.28 to 2.5, a 10 times increase in growth rate because you have so much mixing that you're providing the bacteria these ideal conditions under which to grow. And then the next kinetic rate up from that are complete mix with return activated sludge. And this is you're giving the biology a further boost by returning bacteria from a clarifier to boost the biological community instead of just having it be a once through process. And that brings the kinetic rate up to 5. So now we'll get into the next portion here where we actually meet the bugs in our lagoons. Okay, the first one that we've been talking about so far, and this is the main bacterial community in the lagoon, are BOD eating bacteria. Here we're talking about heterotrophs. They are receiving a large amount of organic molecules from our waste, largely carbon-based, and then other associated molecules. So a lot of food for heterotrophs, which are the bacteria that eat food from the environment instead of making it themselves. So of the bacteria in the lagoon, they commonly come in 3 different types. The first type is free-floating. This is just a bacterial cell all on its own floating around in the water column. And these are great at breaking down bacteria, but they can cause some issues because they are not likely to settle over time, so they can contribute to TSS in the effluent from the lagoon. The next type of bacteria are flock forming. These are not as robust as the flocks that you might be familiar with from an activated sludge operation because those are recycled and they're treated a little bit differently. But flock forming bacteria in a lagoon is a bacteria species that as it reproduces, it creates an extracellular polymer that keeps it tied to other bacteria of the same species. So they form this little raft together. The advantage of that in a lagoon is those flocks are large enough that they will settle out of the water column. So they do their part, they eat the bacteria, and then when they're done, the particle is large enough that it will become part of the lagoon sludge and not go out as TSS. And then the final type are filamentous bacteria. These are bacteria that create long chains of themselves. And these are kind of a dangerous bacteria to have too much of in an activated sludge system because they tend to cause problems with settling in clarifiers, but they're really very benign in most lagoon systems. They're just the third type versus the blob forming flocks and the Lone Ranger free floating. These guys just form long strings. And they're actually very useful as an indicator organism to tell you about the health of your lagoon. So all in all, there's too many bacteria, BOD eating bacteria to list here. They're very diverse, very, very many types. But what they are heterotrophs, and it's a diverse and it's fast reproducing community of bacteria. They're very robust. They're hard to kill. They adapt to changing conditions pretty quickly because if a condition favors one species over another, that species will just reproduce faster and create the bacteria it needs to take advantage of that condition. Next up, we have sludge digesting microbes. So as solids settle out of the lagoon, either near the head of the lagoon or as biomass settles out later in the lagoon, those solids still contain energy. They contain molecules that can be further broken down. And that's what happens in the sludge. And the first type of bacteria that do this, largely in anaerobic or kind of microaerobic conditions are acid producers. From our example earlier in this presentation of a bacteria that creates lactic acid out of glucose, that's a very common thing for many of these acid producers to do. They will take an organic molecule and they're making an organic acid out of it. They'll break it into a couple pieces, and that's how they get their share of the energy from it. Those organic acids then become food for methane-producing bacteria. They will eat those acids. It's a symbiosis where the one bacteria is creating food for the other, and they produce methane. And methane, CH4, will bubble out of the lagoon and out into the atmosphere. So this is a second mechanism besides CO2 production of true CBOD removal from the lagoon. So what doesn't get removed in the aerobic part of the process may be removed in the sludge. And then finally, we have sulfate-reducing bacteria. If they do not have access to oxygen or a better electron donor, they will start to reduce sulfate, which creates hydrogen sulfide, which is the wastewater odor that everyone is familiar with and can become a huge problem if these are allowed to grow and the conditions favor them versus a more neutral, benign sludge production species. So if your sludge blanket is operating correctly, the bacterial activity there will help reduce sludge levels, it'll break it down, and it'll reduce the total BOD. And in a healthy lagoon, the byproducts that are created in the sludge blanket tend to get oxidized as they rise through the aerobic waters above it. That's why if you don't have odor in the lagoon, that doesn't mean you don't have anaerobic activity, but the anaerobic activity is happening far enough under the surface that their byproducts are further consumed and you don't have odor problems or sludge bulking. But if this goes bad, if you've got too much settling and not enough oxygen, you can get severe odor problems, and then you can get things like floating sludge, where methane is being produced out of control, especially during the spring and the fall if you have pond turnover in an inadequately or non-mixed lagoon. And then you get floating sludge, and then that itself creates a new set of TSS and odor problems. Okay, so next up in our review of species in the lagoon is algae. So these are autotrophs. They float on the surface of the lagoon or in the first few feet of water where light can reach, and they're using photosynthesis. They're using sunlight for energy, at least during the day. And in a facultative lagoon, these can be very helpful because they add oxygen in daylight. That's what they're designed to do. But when they are not receiving sunlight, many species can switch their metabolism where they consume oxygen and they consume food from the wastewater lagoon, so they can help drop DO during the night. And algae, they tend not to settle. They can create rafts and float on the surface. So if there's no mechanism to keep them in control, they can create huge problems with TSS, total suspended solids going out in the effluent. And also our second picture here on the bottom right is a type of algae called blue-green algae or cyanobacteria. This one type of algae can cause problems because they do produce toxins, and we'll talk about them more a little bit later. But these are considered blue-green algae as opposed to just green algae. And all of these species, however, have trouble growing in moving water. They are used to operating in still water, so one way to combat excessive algae growth is to add mixing. So moving on to a very special class of bacteria are the bacteria needed for nitrification, to remove ammonia. These are being discussed a lot nowadays as regulators are adding ammonia limits. Lagoons naturally, sometimes, particularly in the summer, remove ammonia on their own, but they don't tend to be very good at it, definitely not as good at it as BOD, because the bacteria are different and they are very not optimal for lagoon conditions. So the primary species we're talking about here are called Nitrosomonas and Nitrobacter. There are other species that do the same things, but these are the two that are most researched, and we design around them because they're the big workhorses. And these are autotrophs. They're unusual autotrophs in that they use chemical energy from the breakdown of ammonia instead of photosynthesis to make their own food. And their growth habit is attached growth. They do not want to be free floating in the lagoon, which can be a problem because compared to the bulk water in the lagoon, there's not a lot of surface area. So they will grow on sidewalls and on baffle curtains and on aeration systems and anything they can stick to in the lagoon. But that's not a lot compared to the actual water volume that you'd expect. And these are autotrophs. They have to make their own food, which is inefficient compared to heterotrophs who just make their own food and can grow much more quickly. So for nitrifiers to survive, they need the BOD to be low enough that they're not competing with heterotrophs for nutrients. And then the last feature of them is that these are bacteria that prefer warm temperatures. They slow down very significantly around 50 to 60 degrees Fahrenheit, and they functionally stop under around 40 Fahrenheit or 4 Celsius. And that can be a huge problem for lagoons in the wintertime. They can still break down BOD even when it gets very cold, as long as the water is liquid. But nitrification, even if you have enough of it during the summer, will completely fall off if your water temperatures start to get low during the winter. So these are just very, very delicate bacteria. They're very much the opposite of your BOD eaters that are so robust. And why is that? This lower energy metabolism. Well, if we look at our electron tower from the beginning, you'll remember that a BOD eating reaction consumes the entire span of the tower. Nitrification is down here. It is less than a third as energy yielding, so they have to eat a lot more to get the same baseline energy. And the reactions just aren't as efficient. And then there's also two reactions. We're going from ammonia to nitrite, NO2, and then further to nitrate, NO3. So it's a lower energy metabolism, and that's part of the reason why they are so delicate. And then as autotrophs, they need a carbon source. They're not getting their carbon from their food, so they need to get their carbon from the water around them. And what they use is alkalinity. So the guiding ratio here is that you need about an 8-to-1 ratio of alkalinity to ammonia to nitrify. And in most wastewaters, this isn't a problem. If you've got hard water, you have alkalinity in the water, but some water sources are naturally extremely low in alkalinity, and that can be a big problem when you're trying to nitrify, because they just do not have enough carbon to reproduce and grow. And then lastly, they have a higher O2 demand. As they do this complex breakdown, they need more of that electron acceptor. So you need to have more O2 in the water, about 3 times as much per pound as with BOD. And then they like DO levels from above 4 milligrams per liter, which you're not going to have without great expense in the bulk water of a lagoon. And then lastly, this does not often happen in lagoons, but it's worth covering as some states are starting to look into rolling out total nitrogen limits. The next step in bacterial treatment is denitrification. So if nitrification was taking ammonia and changing it to nitrite and then nitrate, denitrification is taking that nitrate and turning it into nitrogen gas and releasing it to the atmosphere. Once again, through bacteria, we have true removal from the waste stream. The problem is denitrification bacteria need just about the exact opposite conditions as nitrification bacteria, because denitrification is carried out by heterotrophs, and they need anaerobic conditions because if you have no oxygen in the water, these bacteria that carry out denitrification will use the nitrate produced by nitrification as the oxygen source. It will use it as its electron acceptor instead of oxygen if there's none to be had. So that's the first two differences between nitrification and denitrification. You've got autotrophs instead of heterotrophs. Nitrification, you need a lot of air. Denitrification, you need no air. And that is problematic because denitrification has to take place after nitrification, because if there's no nitrate in the water, the denitrifiers have nothing to work with. So you need to go from a DO of 4 or more down to a DO of less than 0.5. And then another significant difference and a big source of cost when you're doing denitrification operations is that we're dealing with heterotrophs and they need a carbon source. They need BOD, which we've already removed from the water to make sure that heterotrophs wouldn't outcompete our nitrifiers. So if you're doing a once-through process, you need to add some kind of carbon source back in. Otherwise, heterotrophs, these denitrifiers, will not grow. And then lastly, this is a happy side benefit where with alkalinity, that if nitrification consumes alkalinity because it's taken up into the cells, denitrification creates CO2, which puts the alkalinity back into the water. So if you have a very tight pH situation, denitrification can actually help smooth things out if you've got pH fluctuations after nitrification. And lastly, moving away from strict bacteria, we have higher life forms. These are usually multicellular organisms, and they, instead of eating the wastewater molecules, they're eating other bacteria or algae. They're a really robust sign of a healthy micro community, because if you have trouble growing bacteria, you're definitely going to have trouble growing the bacteria eaters. Another nice thing is that under a microscope, they're very easy to see and identify. So we have this concept of indicator species. If you can't identify every species in a lagoon, the ones you can see, you can learn things from about what's going on in the lagoon. So two of the really common ones are rotifers, bacteria eaters, and then the next one is Daphnia, and these are algae eaters, also called water fleas. And we will be talking about them a bit on the next slide. So getting into how do you use all this bacteria knowledge to diagnose problems with the lagoon? If your lagoon is mostly treating its BOD, maybe you don't need to know, but if you're having trouble, bacteria can give you clues as to what's going on in the water. And one that you don't need a microscope or a lab for is the lagoon color. A lot of the color in bacteria is based on a lot of the color of a lagoon is based on the bacteria in that lagoon. And so when that color changes, you can use that as a clue as to what's going on. So one, Daphnia, the water fleas that we talked about last slide, they turn red when there's low DO, when they're stressed out. And that appears in the lagoon as streaks in the water. So that can be a clue that you're having issues with your aeration system or you're having dead spots, is that you've got red streaks in the water. Another one is the blue-green algae that we discussed earlier. That algae likes a low pH. If you've got an overabundance of acid-producing species or low pH in the lagoon from your influent and low DO, you can get a lot of that cyanobacteria, the blue-green algae, and that makes the water look like a dark pea soup. Instead of the very bright green top layer that you see with a lot of algae, this is a duller, darker color than that. And lastly, there are red-purple bacteria that can grow when there's an overload of nutrients with inadequate DO. If you've got a ton of nitrogen and phosphorus, that can be the cause of a red-purple color where your lagoon should usually be blue or green or brown, with kind of clear water with solids being disturbed in it. If it turns colors, that's an indication that your bacteria are unhappy. And then if you do have a microscope available at or near your lagoon, obviously less likely to have a wastewater lab on site than, say, an activated sludge plant. The easiest thing to see from that are higher life forms. You're not going to be able to tell which bacteria is which just looking under a microscope, but you can see your higher life forms. So if you see rotifers swimming around, there's another species called ciliates that tend to be very delicate. If you see a lot of those, you know your lagoon is probably reasonably healthy. But if you're not seeing any of those in the water, that's an indication that something's wrong. They often are very sensitive to toxins. So if you have an industrial plant upstream or something weird is coming in with your influent, that can be an indication that those species will be the first ones to die. So if they're missing, take a look at what's coming into your lagoon. Next up, if you do not have a lab located on site, don't have access to a microscope, which most people don't, you can send off a sample of your lagoon biomass for wet testing. So this is a biological lab that will take a look at the bacteria under a microscope, and they will do tests, such as staining tests, where they apply different dyes to see how bacteria react. And that can help you have a really good indication, a really scientific indication of what is going on in the lagoon. So they'll come back to you with a report that usually includes several sections. The first one will be an assay of the higher life forms in the lagoon. So they'll have a list of life forms they're looking for, and they will make notes whether they're present or not, if they're abundant, if they're very few, which types there are. The next one is an assessment of flock structure. That's the bottom corner picture here. They will give an assessment of how many filamentous bacteria there are in the flocks. Are the filamentous bacteria bridging between flocks? Or are they free floating? How large are they? How compact are they? And then the last one is an identification of filamentous species. Filamentous species are an excellent indicator organism because a lot of research has been done. There's dozens of species and different strains of filamentous bacteria, and each one is associated with a certain condition. So when you diagnose and you see, okay, you have these 3 species of filamentous bacteria in the lagoon, and they're all associated with toxicity, then you probably have a toxicity problem, that you've got something poisonous coming into your influent. I used this in my previous career in industrial waste treatment at a landfill that was very low on phosphorus. They had a lot of filamentous bacteria, and you diagnosed what species you had, and all of the top species, indicator of low phosphorus, indicator of low phosphorus. And that was part of the proof that they needed to bump up their treatment to add more phosphorus to meet those macronutrient needs in the lagoon, because the filamentous bacteria were growing despite the lack of phosphorus, but their overall treatment was suffering. And so beyond that, they can help diagnose a food to mass ratio. That's standard in the reports. That applies more to activated sludge plants, where you can control the biomass in the lagoon. But it's also an indicator, looked at a different way, of how much age you have in the lagoon. So if you've taken a sample near the end of the lagoon and it indicates a lower sludge age or a less robust community than the hydraulic retention time that you think that you have, that might be an indication that you have short circuiting in the lagoon. And you've got older communities in parts of the lagoon, but the bacteria that are actually near the exit might look a lot younger than they should. So lastly, a word about bioaugmentation. There are a lot of companies out there that this is their product, is they will produce bacteria for wastewater treatment operations, and they make a lot of claims about what it can do, and they really can do very interesting things. And two of the most valuable applications are seeding new reactors. Our nitrox reactor that I mentioned at the beginning of the presentation is designed to cater to the preferences and needs of nitrifying bacteria, but they are unavoidably slow to grow. So even when you give them perfect conditions, it can take a long time to fully establish the community in a new reactor. But if you add bioaugment with nitrifiers that were grown in a lab, that can speed things up considerably. Same thing, you can seed a new lagoon with sludge from another lagoon. That's really often done in new activated sludge plants. And then the other application that's really interesting is sludge reduction. A lot of companies will sell a blend of bacteria that are designed to go back and eat any volatile, useful molecules that are left in your sludge, and that can reduce the sludge blanket in a lagoon without needing to do dredging. But it needs to be said that bioaugmentation is not a silver bullet. If you're tempted to think, I don't like my microbes, they're not healthy, I will just add new ones. The new microbes are going to fall victim to the same conditions that affected the old microbes. So if you have unhealthy conditions, adding new microbes is not going to fix it because they will still die. You need to fix the underlying causes. And okay, sorry about that. New web software. So I have a question from Josh about spring turnover. Does it occur because of cold temperatures over the winter? Your understanding is that the bacteria wake up in spring and quickly break down food. I have not heard that explanation for it. The traditional explanation for it is the bacteria might assist with that process, but it's largely a function of temperature change, is that you've got a layer of colder or warmer water on the top of the lagoon where it's exposed to the environment, and then you've got a layer of the opposite temperature that's either cooler in the spring, it's not warming up as fast, or warmer in the fall. And those layers, if you don't have mixing, will stratify, and that's how it does. You get lagoon turnover. I've got one asking about mosquito problems. So mosquito larvae also really do not like moving water. So if you add mixing, that's one way to stop them. And then if you have a robust community of higher microbes, some of them will actually eat the mosquito larvae. From Stephen, we have a question, how do you create a favorable anoxic zone in a lagoon? A lot of that is the design of the lagoon based on depth. The anoxic zone should really just be in the very top layer of sludge. So if you have adequate aeration and mixing above the sludge layer, you're going to have a much healthier sludge blanket that's not as thick, you're not over settling solids, and that's going to create a much healthier anaerobic layer. We have a question about, does the triple point Mars aerators assist anaerobes with reduction of lagoon sludge blanket? That actually is an excellent question. It does, but it's not the anaerobes that do it at that point. Our Mars aerator keeps so many sludge solids in suspension that they actually are moving through the aerobic layer, and they're much more broken down in the aerobic part of the lagoon. So yes, it does help with sludge because it's got so much more mixing, but it's not anaerobic at that point, it's aerobic. We've got a question from Darlene. Is it possible to have anoxic and aerobic conditions in the same tank? Absolutely. You get stratification. This is how a lot of facultative lagoons are designed. Your aerobic conditions are near the surface. You can also get this with surface aerators in deeper lagoons if the air doesn't penetrate down to the bottom of the lagoon. So you can have a huge layer of aerobic water sitting on top of an anaerobic layer of water and or sludge. We've got a question from earlier in the presentation. Isn't complete mix another term for activated sludge? It is functionally the same level of mixing as activated sludge, but the activated part of activated sludge, you need a sludge return for that to work. So we differentiate between complete mix, return activated sludge only when you're recycling microbes. If it's just complete mix, then it's still lagoon treatment. It's just a very, very aerated lagoon treatment. We've got some questions about the slides. I believe this webinar is going to be put on YouTube in the future. I have a question about thoughts on phosphorus removal, and I wish I had a better answer for this. Biological phosphorus removal is really difficult in lagoons. It can be done biologically, but there's no gas form. There's no way to discharge the phosphorus to the atmosphere like you can with carbon and nitrogen. So if you're doing biological phosphorus removal, EBPR, enhanced biological phosphorus removal, you can trick a certain class of microbes into accumulating phosphorus in their bodies. But then you still have the phosphorus in the system unless you physically remove that sludge. Then you're just locking phosphorus into the sludge. And if you have turnover in the future or an upset, that phosphorus is not actually removed from the system and it might come back to haunt you. That was from Shiloh. We have a question from Josh about can filaments be found in oxic, facultative, and anaerobic conditions? I believe filaments are most common in oxic and facultative conditions. I do not know off the top of my head if there are anaerobic filaments, but I would not be surprised if there were, because they tend to be a class of bacteria that they grow quickly and they just try to exploit gaps where other more robust bacteria can't grow. And it is 3 o'clock. I'm sorry for anyone whose questions I have not gotten to. And if you have any questions, please let us know on our blog or by email. And we should be putting this webinar on YouTube, and we will be monitoring the comments there. So thank you very much, everyone.